Can everyone see my slide? Okay.
Now, yes, full screen.
All right.
Okay.
Oh, thank you, Sebastian, for organizing this. This morning, I was very happy to hear the talk of Michael Levitt, because he was also talking about simple statistical models can represent very complicated COVID data with so much of error and variability, but still the average data can reflect some underlying statistical principle. So in a similar light, my talk is actually tuned towards that. Can we see some statistical laws, simple laws in omics data? And how does this data can help us to do a better, higher resolution data analytics? So before that, I'd like to introduce my team members here. So the core members constitute of Mohammed Helmi, is working on largely omics data. Then we have Hock Chuan, who's working on machine learning models. And Olga is working on data analytics, also on transcriptomics data. And Jasmeet is in SIPB, but because I have a joint appointment in SIPB, and she works on dynamic metabolic modeling, doing experiments as well as modeling. And some of you have heard her talk before. And also I have co-members, joint PhD students coming on board and interns as well. Okay, so our group largely focus on two areas. One is the dynamic modeling, looking at signal transduction, metabolic network, trying to validate the model with time series data, as well as space-time model if such data are available. On the other side, we also look at, as I mentioned, data analytics. And today my talk will be more on this side. And here I'm just showcasing on the right side that we have developed tools that can actually go through the entire process of RNA-seq analysis, for instance. So we can import RNA-seq data directly from the GEO database. We can do pre-processing, and then we have multiple functions, data analytic functions, correlation, noise, PCA, t-SNE, SOM, and also finally the gene ontology part, try to find the protein-protein interaction and so on. So you can check it out. This is our new web server. It's free to use, and it doesn't need any programming skills. Okay, earlier on, sorry. Earlier on, Michael did mention about large-scale network and power law. And this is an example that we can see visually, right? If you look at the airline network, the one left in the United Airlines, and you see a lot of interconnectivity, but you will also see hubs such as New York, San Francisco, these are hubs. And likewise in social media as well, whether it's Twitter, Skype, or Facebook, you will also see some individuals who are very famous, highly connected, and then you see individuals that are not so connected. And this can be represented as a scale-free network. And theoretically, Barabasi and his team have done significant work. He's a physicist who looked at all sorts of networks, social network, biological networks. And what they observed studying this network, most of these networks are following scale-free, as I shown earlier. We have hub and you have nodes. That means the hub are the ones that are the key. And this is important for biology because when they talk about essential genes, or when you want to target diseases, you probably want to look what are considered hubs. So this has been an intense area of research for some time, trying to use statistical laws or network to understand key properties or key proteins or genes in biological networks. So I won't talk much about the details because Michael already given a very good example. So simply, a hub is, as I said, is connected to other nodes, and the nodes have very few links. Hubs have many links, and you can follow a relations that follows the power law. And this power law has been observed in several species. And here's one paper back in 2004 studying microarray data, and they're looking at E. coli, yeast, and various species, including mammalian cells as well. And they see this nice distribution of genes following more or less power law. Okay, and we also investigated this power law distribution across different cells using RNA-seq data. And we were looking at across different cell size. I mean, from single cell to a cell population. And you can see whether it's a single cell or a population of cells, with 100 cells here, or even 10,000 cells, they have similar exponent, power law exponent, which is roughly about 1. So generally, they follow power law. So this has been observed in the data. So what is that this data is following power law? So we wanted to understand deeper why, because if we know the statistical structure better, as I said earlier, we may be able to find higher resolution of data that are valuable instead of just looking at the highly expressed or highly changing genes. And normally biologists take a handful of them, sometimes even 5 to 20 or even 100 out of maybe 30,000 genes. So we want to go deeper to see the statistical structure. And here I just have distributed across different cell type between the replicates. I mean, gene expressions across replicate in a log scale, from low to high expressions. And what we can see is it's more or less a linear line, showing that these data are highly correlated, but there are variability in certain gene expression. And you see more variability in cancer cells on the right bottom. There's more kind of differentially differential expression even between the replicate. Whereas if you look at more the microbes, they are less variable. So knowing this information, this kind of scatter, how can we look at individual data and make inference for each one of them? So how can we select effective set of genes considering this noise or variability in order to make a better judgment of the data? So before I go into that, I want to introduce this noise. So noise is simply variance divided by mu squared. So it's or in other words, coefficient of variation squared. That's noise today, we know, and we can use that expression to calculate this for individual genes, and then you sum them to get a total average noise. And you can see that many of these differentiated cells, the noise varies between 0.6 to 0.8 units. Whereas for the developmental cells, interestingly, there's some noise at the earlier part of the cells are very low, but later on they increases. And the story for this is because the nucleus has not been formed. Transcription caused much of the noise, transcriptome white noise, and this was not just available for up to 2 cell. So you can calculate total noise as well as you can calculate noise by sorting according to the expression. So you sort the expression from low to high and you calculate the noise, and you can see that noise actually scales down when you go high expression. And the way it scales down can be different between different cell types. So using such information, noise and variability, we want to understand each individual cell behavior, cell type, or even conditions. So here, theoretically, we want to first understand the distribution. So here we artificially created transcriptome-wide data using a Poisson distribution, which represents more or less stochastic noise across a gamma distribution of single cells known to follow some gamma distribution. So we take gamma distribution and also with Poisson, and we get this sort of behavior. On the left side, we can consider it just with the stochastic noise, that means intrinsic noise. And when we take 30 samples here, 30 times we distribute this, and we take the average of it, the noise reduces. And if you look at the noise across the expression, it follows this curve that is initially for single cell, the noise is high, but as you increase the expression, the noise reduces. And for average, you have overall less noise. This is an artificial theoretical distribution, has no real meaning, but we want to use this to understand real transcriptome-wide noise. And here we artificially added what we call variability, that means extrinsic white noise. So we just randomly add white noise into the original data here, and then we can see that now you have more scatter. The correlation drops. Here it's pretty high, here it drops. And even when you take the average, you have the scatter disappears because it's just plus and minusing of the noise. So you have a less noise when you look at the population. And when you look across the expression, again, you start with a high noise at a low expression, but this time it saturates around some number. So it's asymptotically going to some number, and that's because of the extrinsic white noise. Now, how does this compare with real experimental data? And then you can see the real experiment data. This is for mouse oocyte. It's kind of between these two situations, right? And it's kind of a mixture of these two. And using this kind of laws, you can actually find the underlying noise structure for each kind of cell. And the two things we can see, that one thing is that the population average, you have less noise, and also at expression level, you have less or close to zero noise when you go at high expression. And we studied the same data just now I've shown you. We saw the scatter plot here. And then when we look at the noise, we see the noise are generally going down. But then the dotted lines represent the law of large numbers. And it's a statistical law, how, when you sample many data, how the noise will reduce. So from this data, one thing we need to know, we have not done any expression cutoff. So we are using all data. But we know that low expression genes have noise, and this noise we need to eliminate. So here we are using some, again, distribution plot over the whole transcriptome-wide data. We make several distribution. Earlier on, I talked about power law distribution, which is Pareto. And we tried different distribution for different kind of cells. So we have E. coli, yeast, and here we have cancer cells, and then fallopian tube cells. So when we use this different statistical distribution, overlay with the real data, what we see that upon a certain threshold value, you see that the real gene expression matches many of these distribution data. And then if you use the ARIA-K information criteria, then you may be able to pick up the best fit. So for this data, this is cancer data, or most data, in fact, we are getting log normal distribution. I mean, the gene expression follow log normal, and then we're using the particular curve for the log normal, we can find the intersection where the line kind of deviates from the experiments, and we use that as a lower expression cutoff. So initial data was no cutoff, which is here. And when we use the lower expression cutoff using statistical distribution, we can see that the noise scaling with the number of cells here is following the law of large numbers. And we also see such law of large number law or inverse square law in different settings and different kind of data. I mean, here's like looking at the standard deviation of Pearson correlation, standard deviation mutual information. This is for E. coli data. And even for cancer data, we see some sort of distribution that's going asymptotic and following more like a law of large numbers. So how is this information? So in this case, for example, as I said that this noise saturates at some value, some asymptotic value. So we can use this level as a means to cut off. So it saturates here, so we can take a cutoff in this region, and all these genes expression we can remove as one way to see how the whole overall transcriptome response behave. So for example, here it's going back to the cancer data, ovarian cancer data, and we have done SOM, self-organizing map of the top 500 genes. So we just look at the top 500 expressed genes, and we try to generate SOM. And we can see that there's no much, I mean, there's some kind of pattern, but it's not very clear. And here we use the whole data set. And when we use the whole data set, we tend to see some patterns arising. All the FTP are grouped together, and then the cancer cells, there are two groups that are formed. Now we use the noise cutoff, the one I showed earlier on. When we use this noise as a cutoff, we have 8,600 genes now instead of 18,000. And now we can see the resolution is higher. So for the FTP, you get almost very nice distribution among the different individuals, fallopian tube. And here for the different patients, we can see that two major groups are happening. So even though they are from a subtype of high-grade ovarian serous cancer, this subtype, based on 6 patients, can be divided into 2 subgroups by this SOM plot. And again, we use this statistics and noise analysis. We have used this to come up with a new way to select differentially expressed genes, and this is called SCATLay. So what SCATLay does is a tool that can overlay your replicate data as well as your condition data, that means wild type and mutant, for example, versus your replicate data. So what it does is subtracts whatever replicate noise that you will see on the condition data. So that means the yellow ones will be removed and what you have is a green one. So when we do that and we compare with the very popular differentially expressed genes tools like DSeq2 and NoiseSeq2, we can see that SCATLay has higher coverage. It has more coverage than the others. But of course, if one has too many genes and they would not want to analyze all the genes, they're only looking for subset, and you can still create threshold to reduce the number of genes. You can use, for example, fold change values, or you can use expression values to further cut down. But if you don't want to cut down, you have a higher number of genes for analysis. And using this sort of tools, noise, statistical distribution, we want to go further in our lab to understand more complicated data that are arising. Now we have spatial transcriptomics data, and we also have spatial metabolomics data. And there are connectivity, right, between the genomics, proteomics, and metabolomics, but these connectivity are not well studied because they are too complex. And adding to this is this spatial and temporal data. So clearly, using just statistics, simple statistics alone is insufficient. So now we are exploring more like a machine learning type of approach, how we can create machine learning models that connects the dynamic or spatial gene expression data versus the metabolomics data, for example. And this is a complex thing. And often we don't have enough data to create this sort of model. So we are now in the process of trying to generate artificial data to actually come up with the initial model before the real data comes. So in summary, so I just want to highlight the gene expression follow power law or scale-free network or log normal distribution when you look at the individual expression. And this noise reduces similarly to the law of large numbers. And using this distribution fitting and SCATLay, we can get wider number of genes for analysis. And in future, these statistical laws can be useful for generating synthetic data for machine learning applications in biology. With that, I'd like to thank you. And also like to thank the following institutions that we have collaborated and still working. And in particular, I'd like to thank two of my ex-lab members, Vincent and Tutin, who have worked a lot on this data that I presented today, and Sandro for being a good collaborator for many years. Thank you all for listening.